I'm David Fay. I'm the proprietor of the Wyoming Worm Lab since 2001. This was given to me by a good friend of mine. Okay, so what we do here is we basically use nematodes to try to figure out how things work. That's what sort of gets us excited up in the morning, and just trying to understand fundamental biology. A lot of it is related to human disease and health, but mostly it's puzzle solving. So the worms we work on, they live on these little petri dishes. This is not their natural environment. You could normally find them out in the soil, but instead they live on these things, and there's like a lawn of bacteria, so they get to like crawl around in their food and eat and reproduce. And you might ask, why would you study a nematode, right? They share in common many, many genes with humans and with all species, and that's the basis of evolution. You invent something once and you keep using it, and it gets tweaked and it's slightly different when you go to different organisms. The thing about worms that's great is that you can essentially figure out the answers to your puzzles in a lightning fast way. So over the years, we've sort of worked on many different things, and part of the way we work is we sort of let the research lead us where it's going to go. You have some control over it, but in the end, you want to follow the things that are most interesting. Basically, what you do in genetics is to figure out how something works, you break it. And in the field of genetics, the way you break it is you introduce random mutations, and then you look for very specific defects. So if you want to know how an organism forms its head, you'll mutagenize them, and then you'll look for animals with 2 heads or no heads, or 1 head but it's all messed up in some way. And what you do is you get these mutants, and then we use methods like whole genome sequencing, for example, which has totally revolutionized the way we do our work. And you find which gene is the causative mutation in your background that led to that defect. And you can get dozens or hundreds of such mutations, and you can really piece together a whole molecular genetic network to figure out how something works. And then what you're always hoping for, and it's true more often than not, you're getting genes that are highly conserved in humans. Often genes you're coming across have been implicated in disease, but no one knows really what they're doing or how they're working, what is the function of the proteins they encode, and that's the kind of thing we can figure out quickly here, relatively quickly. But I guess the steady theme in our lab has been understanding how lots of different mutations interact with each other. So we all carry tons of, you don't necessarily want to call them mutations, they're just variants. I'm different from you, we're all different from each other, and that's because our DNA is slightly different. It's 99.99 something percent identical, but it's the differences that make me, me, and you. So understanding how, this mutation in this gene and this variant here and all these different things come together and create this precarious balance that can lead to a disease or maybe a predisposition to a disease is something that we've always been interested in. And we've tried to pioneer sort of genetic methods to study that in a much simpler organism, the worm. A lot of what I feel like is exciting about working on microorganisms is that they're sort of invisible to the naked eye. What's fun is you put them under here and you look and you're like sucked right in. A lot of it's observation. We do a lot of molecular stuff, we do a lot of genomic stuff, we do a lot of stuff with fancy equipment, and this is pretty basic. But at the same time, if you can't look and see things, you're just going to miss stuff, and that's how science gets done. You just look and you notice things. I think you have to love this. That's the first point. You have to have the intellectual curiosity, the deep curiosity that sort of really, you can't wait to get into the lab in the morning to like look and see what happened. You also have to have a lot of stubbornness, because most of the time you're disappointed, right? You do it, it's like, but you have to really have the guts for science. You have to have that stubbornness to keep plowing through. I think one of the things that has really made this career worth it to me is the people. In fact, probably more than the science itself. I like people who love science and who are dedicated to this, semi-obscure at times pursuit of truth and knowledge. I enjoy having undergraduates in the lab a lot. And I think the undergraduate experience is really important because, some of them discover this is it. I found my tribe. It turns out I'm a scientist and this is what I'm destined to do. Many others, maybe the majority, discovered, this was interesting. I have an appreciation for what scientists do now, but it's not for me. I need more instant gratification. I need to have that feeling of having helped multiple people every day or accomplished little tasks. And that's a really important, valuable experience too. I don't care what people use their degrees for. their graduate degrees, their undergraduate degrees. As long as this is serving them and it's helping them move on with their careers and lead a fulfilling life, then I'm happy. So how long does it take to actually get things done? The answer is much longer than you always expect it to. Although occasionally things can happen really quickly and you're just racing to keep up. The overall arc of the lab, I don't even know what that is yet, right? We're just sort of midway there or something. But projects ebb and flow, but they often go on for years, many years, and multiple people really busting it to get it done. And it's like getting a fish on a line. That's the best example I can, sometimes you're out there, you're casting forever, you're waiting forever. And the bottom line though is, as soon as you've got that thing, you better set the hook. And that's where it really comes in. Once you've got something, you just go after it with a lot of tenacity. You just don't let go. You don't let that fish get off the line. You keep reeling it in and you do whatever it takes to get it in and to make the story, to figure out the piece of the puzzle.